Hello students. In the last lecture we had talked about the primer design. So what I had talked about is that how you can design the sequence of your primer. Okay? आप अपने प्राइमर के सीक्वेंस को डिजाइन ही कर सकते हैं, सिंथेसाइज नहीं कर सकते. क्यों? क्योंकि सिंथेसिस के लिए हमें उसे भेजना पड़ता है. कहाँ भेजना पड़ता है? We have to send it for synthesis to the companies. Why? Because those companies who are capable of having the infrastructure or the instruments required to carry out the chemical reaction for the synthesis of those oligonucleotides, those companies will make the primer for you or oligo for you, and then it will send you that chemical molecule. You can only tell the sequence of the chemical molecule depending upon your requirement. So therefore, our next topic is synthesis of oligonucleotides. Dear, the synthesis of oligonucleotides, before I talk about it, so the term oligo means a few. Few means between 2 to 10. But generally, oligonucleotides, they are synthetic molecules which are synthesized chemically in the industry, and they can be anywhere between 2 to 150 bases. So we can say that these are short single-stranded DNA or RNA molecule that have a wide range of applications in the molecular biology, genetics, research, and forensic. Okay? And primer is one type of oligo. All oligos are not primer, but all primer are oligos. So we need the building blocks for the synthesis of oligos, and the building blocks would be, for example, for DNA it will be deoxynucleotides, for RNA it will be ribonucleotides. Okay, so similarly there are different types of nucleosides which are required for the synthesis of oligos, and they can either be obtained from the natural resources or they can be obtained from the synthesized chemically during the chemical reaction. And once these oligos are formed, then they can be joined together to form the linkages leading to the formation of the oligos. Before I go further, I want to make you people familiarize with a few terms. ठीक है, आपको कुछ चीजों का, कुछ terms का पता होना चाहिए. And these terms are such as protecting group. So why I'm discussing about these terms is that these terms will help you in understanding the process of oligonucleotide synthesis. Okay, so what is a protecting group? A protecting group is, now, are the chemical groups which are present at a particular position. Now, why they are present at a particular position? They are present at a particular position because they prevent the unwanted chemical reactions and therefore allow solubility in the ऑर्गेनिक सॉल्वेंट्स. मतलब अगर आप चाहते हैं कि आपका कोई भी मॉलिक्यूल है, चाहे न्यूक्लियोसाइड है, चाहे न्यूक्लियोटाइड है, आप चाहते हैं कि इट शुड नॉट रिएक्ट विद एनीबडी, and there are certain reactive atoms which are present on its surface, then you would block or you would stop the unwanted reaction by adding these protecting group. For example, here you can see that in case of adenine, cytosine, and guanine, there is amino groups are present. Okay, adenine, cytosine, and guanine, they have amino group, and these amino groups are very reactive in nature. So in order to prevent the chemical reactions of this amino group, you can see that certain chemical groups are added to the adenine, cytosine and guanine, such as BZ, that is benzoyl group, isobutyryl IBU groups. Okay, so they are added to the different types of nucleosides. Why do they are added? Because they make them stable and they prevent unwanted reaction. However, these groups, they can be removed once the oligo is formed. ओके, तो हो क्या रहा है बेटा कि जब भी हमारे पास न्यूक्लियोसाइड हैं या न्यूक्लियोटाइड हैं और हम चाहते हैं कि उनकी अनवांटेड रिएक्शन ना हो, सिर्फ हम क्या बनाए, आपस में फॉस्फोडाइस्टर बॉन्ड बना के एक आपको न्यूक्लियोटाइड सीक्वेंस दे दे या ओलिगो दे दे, तो हम नहीं चाहते कि इसका OH ग्रुप या इसका NH2 ग्रुप या कोई और ग्रुप है वो किसी और से रिएक्ट करे, तो हम उन ग्रुप्स को ब्लॉक कर देते हैं. ओके, वी कॉल देम एज प्रोटेक्शन. Okay, and those groups which make bond with such reactive groups or reactive positions, they are called as protecting groups. Okay, now similarly, जैसे मैंने बताया अमाइनो ग्रुप को ब्लॉक करने के लिए BZ या IBU है, सिमिलरली 5 प्राइम OH पोजिशन जो है उसको भी ब्लॉक करने के लिए, दिस इज द 5 प्राइम OH पोजिशन एंड इट इज वेरी रिएक्टिव इन नेचर, इसको भी प्रोटेक्ट करने के लिए a group called as DMT, which is dimethoxy ट्राइटाइल. ओके, ये ग्रुप ऐड कर दिया जाता है एंड यू कैन सी दैट दिस इज डाइमिथॉक्सी ट्राइटाइल. ये ग्रुप OH के साथ रिएक्ट करके यहाँ पर अब इसकी फर्दर रिएक्टिविटी को बंद कर देता है. सो ये क्या है, लेकिन ये ग्रुप क्या है, एसिड लबाइल है, मतलब अगर एसिडिक मीडियम होगा ये निकल जाएगा बाहर. It is a temporary blocking molecule only and it can be removed at the beginning of synthesis. ठीक है, जब भी हमने ओलिगो बनाना है तो हम अपनी मर्जी से इसको हटा सकते हैं, जस्ट एसिडिक मीडियम क्रिएट कर देंगे और ये रिमूव हो जाएगा. ओके, सो फर्स्ट थिंग दैट आई हैव टोल्ड यू इज अबाउट द प्रोटेक्टिंग ग्रुप. सेकेंड थिंग इज RNA के केस में क्या होता है, विच इज डिफरेंट फ्रॉम DNA, is that RNA has an extra OH group. ठीक है, अब ये एक्स्ट्रा OH ग्रुप कहां पर है, यू कैन सी ओवर हियर. DNA में एक OH यहाँ था, एक यहाँ था, और लेकिन ये जो एक्स्ट्रा OH ग्रुप है विच इज ऑल्सो रिएक्टिव इन नेचर, इट इज देयर इन द RNA. तो इसको भी, therefore RNA requires one extra protective group as compared to DNA, and R here stands for this protective group. और R क्या हो सकता है, R ये भी हो सकता है, R ये भी हो सकता है, R ये भी हो सकता है. Different companies, they supply different types of protective groups. Okay, so आपको याद रखना है कि widely 3 types of protective groups are used for RNA apart from the common protective group which are generally used at the 5th OH group. ठीक है, so that is for RNA. Now इसी तरह से there is a possibility कि जब चेन बने ओलिगो की, जब सिंथेसिस होने लग जाए तो उनके जो फॉस्फेट बॉन्ड्स हैं, ओके, फॉस्फेट ट्राइएस्टर बॉन्ड्स हैं, ये वाले जो बॉन्ड्स हैं, ठीक है, दीज फॉस्फोडाइस्टर बॉन्ड्स जो हैं ये भी फॉस्फेट लिंकेजेस, इनको दे आर आल्सो रिएक्टिव इन नेचर, सो इनको भी प्रोटेक्ट करना है. सो दे आर नॉट प्रोटेक्टेड एज डाइएस्टर, रादर दे आर प्रोटेक्टेड एज ट्राइएस्टर ग्रुप. ओके, और यहाँ पर दे फॉर्म अ लिंकेज विद 2 साइनो ठीक है, CE. You can see that this is ethyl group CH2 and this is cyanide. So 2 cyanoethyl group है, it protects the phosphate linkages. तो अभी तक हमने बेटा क्या देखा कि हमारे पास प्रोटेक्टिव ग्रुप होते हैं. प्रोटेक्टिव ग्रुप वो ग्रुप होते हैं जो जाके रिएक्टिव ग्रुप्स जो हैं उनको साथ बाइंड करते हैं, so that any side reaction ड्यूरिंग द ओलिगोसिंथेसिस इज प्रिवेंटेड. DNA के केस में 5OH के साथ प्रोटेक्टिव ग्रुप लगते हैं, ठीक है, एंड सिमिलरली फॉर साइटोस एंड एट द सेम टाइम वहां पर अपार्ट फ्रॉम द 5OH The amino group are also blocked by different types of protecting group. Okay, but in case of RNA, one additional protective group is required at the third position because there is an additional OH, and the phosphate diester bonds are also protected by the formation of or addition of a 2-cyanoethyl group. Okay, so these are very important. अब आते हैं हम ओलिगोन्यूक्लियोटाइड सिंथेसिस पे है। देखो बेटा, ओलिगोन्यूक्लियोटाइड जो सिंथेसिस है, it is done by companies or it is made by some chemistry wale laboratories, and there are so many methods which have evolved over the years, but the most common method which is used is the solid phase chemical synthesis, okay, which is represented over here. So this is the most common method of synthesis of the oligo. Okay, now what happens during this process? So solid phase synthesis strategy was put forward by Robert Merrifield in 1962 for synthesis of peptide, but it is equally applicable to DNA also. इसमें क्या किया था कि हम एक बेस बनाते हैं. बेस means this is the background जो पीछे दिख रहा है. This is the background. A background or the base of insoluble polymer, या तो पॉलीमर होगा या सिलिका होगा और ग्लास बीड. It is made as a base or as a solid support. Is support on this surface, what is synthesized? The oligos are synthesized. Okay, so क्या होता है? Products को covalently bind करते हैं. You can see that here. If I give you just an representation, this is any type of P is for polymer. Okay, so any type of polymer is there, and then on its surface the synthesis is taking place. So what is happening is that covalently the products are attached. Say N1 is getting attached, and then the leftover molecules are N1 are washed away. After that, another molecule comes and binds to N1. Okay, then another molecule comes and it binds, and so on. When this is removed, the other one gets washed away. So for this, in 1984, the Nobel Prize was given to Robert Merrifield, okay, for this process or for designing this solid phase synthesis strategy. And this process is, the advantage of this process is that the purification is very easy and it can be easily automated. Okay, so that is the very clear advantage of this method. So this is Robert Merrifield. This is the peptide synthesizer in his lab, and he was from the Rockefeller University. Okay, so this is another representation again of the solid phase synthesis strategy, showing how nucleotides can be synthesized. For example, this is the silica bead, which is used as a solid surface for synthesis. And you can see that this is a T oligonucleotide which is attached over here. Now what happens is that as I have already told you that सबसे पहले हर एक ओलिगो के डीएनए के केस में 5 प्राइम का जो OH है वो ब्लॉक प्रोटेक्ट हो जाता है. कौन प्रोटेक्ट कर रहा है? It is the DMT which is protecting it. So first, if it has to bind with A, then this DMT is removed. इसको हटा दिया. Then the A comes. A has a phosphate on one side, and at the same time A has a blocked OH on the 5